welcome back to the 2001 NSDA Virtual Symposium. We're so pleased that you could join us today, and we have an amazing panel to talk about possible new future treatments for spasmodic dysphonia and possibly other related voice conditions. So once again, I'm going to turn it over to our NSDA president, Mr. Charlie Reavis.
Thank you, Kim, and warm welcome to all of our panelists. And again, participants, have your questions ready. And we had for our panel participants here, just so you know, in the last panel, we had quite a few questions asking us about new treatment options and what might be coming along. So we asked them to hold those for you guys. So what I want to do is ask each of you to introduce yourself, and I'm going to ask the researchers to take 2 or 3 minutes and talk a little bit about your projects, what you're working on, and Risa, if you can talk a little bit about your experience of being a volunteer in one of these research projects. So we'll start with Divya.
Did you say me?
Yes, ma'am.
Can you hear me?
Yes.
Okay, thank you. So I'm Divya Bhaskaran, and I am a postdoc researcher at University of Minnesota. I work with Dr. Jürgen Konchak, and we are working on research treatment options for spasmodic dysphonia. And we have 2 studies going on. One is a clinical trial looking at long-term effects of vibrotactile stimulation of the laryngeal muscles. So the treatment option we're looking at is applying vibration to the laryngeal muscles to see how that changes the voice quality, and also we're looking at cortical activity as well to look at whether the changes in voice quality is long-term, short-term, and we have the clinical trial, which is a long-term study, which is an 11-week study, and we send people home with the vibration device so they can apply the vibration themselves at home. And then we have another study which for abductor dysphonia, and that is a short-term study, and that is being sponsored or funded by NSDA. And we are looking at the same vibration device, but it is embedded in a collar type of strap that you can just strap on. And we're trying to look at the portability of the device and the usability of that device. And so that study is focusing in people with abductor SDA.
Good. Thank you, Divya. Michael. You're still muted, Michael.
How's that?
There you go. Good.
All right. Thank you for inviting me today to the symposium. I'm looking forward to the panel. I'm a laryngologist. I'm the chief of laryngology at Columbia University Medical Center, and I direct the Center for Voice and Swallowing there. And we've been studying laryngeal dystonia for a long time now. The primary research that we're doing is based on electrical stimulation for neuromodulation of laryngeal dystonia. And it appears that, this is something that hopefully will work. It does definitely improve patients' voices. We have to figure out exactly where to stimulate people and how to stimulate them. And we're doing this in consult with Medel Corporation, who's very invested and really believes in this technology for vocal dystonia. Currently, we have our second phase trial going on in Europe, where we have 4 centers collecting patients, and we're doing stimulation of the anterior branch of the superior laryngeal nerve, which is the sensory nerve of the larynx, which carries some proprioceptive messages back to the brain. So if we can manipulate those proprioceptive messages, we might be able to decrease the hyperexcitability of the cortex and mitigate the symptoms of laryngeal dystonia. And that's happening in Berlin, in Vienna, in Jena, and one other city that I'm forgetting right now. So we're recruiting 20 patients, and so far we have 10. In addition to that, an offshoot of this study was trying to come up with some better outcome measures for patients with laryngeal dystonia, and this was funded by the NSDA and by Cure Dystonia Now. And we found something called the Communication Participation Item Bank and validated that for laryngeal dystonia. So it actually is scientifically valid at really the most modern psychometric and mathematic levels that we have for outcome measures. So it's valid for laryngeal dystonia, and it can tell the difference between patients pre-treatment and post-treatment with botulinum toxin. Therefore, we can use that as an outcome measure really for any of the treatments that we're discussing today in the future to see, do they really help? So it's in addition, because we all know that the outcome measures we have for laryngeal dystonia are not very specific at this point. So that's where we're at.
Good, thanks, Michael. Chris?
Charlie, it's nice to see you again, and thank you for the invitation. It's a pleasure to be here. My name is Chris Honey. I'm a professor of neurosurgery in Vancouver, Canada, and I'm very interested in the neurological cause of dystonia. Spasmodic dysphonia is one of the focal dystonias, and we have a track record treating other focal dystonias directly at the source in the brain because it's a brain problem. And we have done a trial using deep brain stimulation to block dystonia, sponsored by the National Spasmodic Dysphonia Association. And that talk is pre-recorded and available from Kim on YouTube. What I'd like to just echo, I think Dr. Bonnie Smith said this earlier, and that is there need to be multiple treatment options. If you can fix your spasmodic dysphonia with vocal coaching, hallelujah. If Botox works for you, hallelujah. But there are patients who have problems with the current treatments, and what options do they have? And so one of the reasons that's one of the reasons that drove us to try and go to the source of the problem and block the cause of dystonia rather than just cover up the muscle dysfunction. And so we wanted a treatment that would go to the cause of dystonia, be one treatment that could treat all the kinds of dystonia, adductor, abductor, mixed, and potentially be a cure because you're not born with spasmodic dysphonia. You develop it from a basal ganglia disorder. And we have one patient who is 10 years out from stimulation who, when we turn the stimulator off, their voice is normal. So we're not sure if this is going to turn out to be a cure, but we're interested in, one, treating the condition, particularly for those people who don't have other options, but also as we are treating the condition, we're interested in learning what is the cause. Because if we can figure out the actual cause, maybe there's a vocal therapy or a pill that will treat this in the future. So that's my introduction.
Great, thank you, Chris, appreciate it. Russell?
I think you said me. My audio is a little bit choppy. I hope you said me.
Yes, you did.
Okay. Thank you again. Thank you guys for inviting us. This is a really great honor to be able to talk a little bit about our research. I'm here, I guess, as a proxy for Dr. Kimberly in her brain recovery lab. I am her postdoctoral student. I've been here at Massachusetts General Hospital for about a year. In the brain recovery lab, we also use non-invasive brain stimulation as a potential treatment for certain focal dystonias. And traditionally, Dr. Kimberly has worked with focal hand dystonia and also AB and AD laryngeal dystonia as well. And with the gracious support of the NSDA, we've been able to run a few pilot studies using repetitive transcranial magnetic stimulation, which is RTMS. And we found that a single session of this repetitive TMS has been effective in improving voice quality, and we believe that's because we're kind of what Dr. Honey has been saying. We're kind of modulating the source of the issue, and we're increasing the amount of inhibition in individuals with laryngeal dystonia to hopefully improve that voice quality. And our most current study that we're recruiting for is looking to expand on those pilot results that we've received. And we're looking to do a 5-day trial of our TMS, so increasing the dosage of the TMS that we're delivering to the motor areas of the brain responsible for voice production. And we're hoping to see both larger effects as well as more long-lasting effects. And we're really, really excited to be recruiting for that. You can get into the study by talking to me. Is it okay if I answer this question that just came up? I'd
Sure.
be happy to talk to you. I don't know if this is appropriate. I don't really care about spam or anything like that. Can I send my email in the chat? Is that okay? Not that anyone would spam me, but
if you choose to, Russell, that's fine.
I choose to. I'll send that. Thank you again for letting us be here and for all the support as well.
Right.
Thank you.
Yeah,
And
I would, Russell, I think too, if you would just take a second and comment that originally or initially the work you guys and Dr. Kimberly was doing was focused on ADSD. And this latest pilot project that you're just starting to kick off and hopefully start to recruiting pretty soon is focused on ABSD.
Correct. So all laryngeal dystonia, all spasmodic dysphonia. So that initial pilot study focused on ADSD because it was the population most readily available to us as it is the more common. And we work directly with the Boston Voice Center here at the MGH Voice Center, and a great group to work with. And so we're hoping to recruit many more ABSD folks as well.
Good, super. Thanks, Russell. Risa?
Hi, thank you for having me. I have abductor dystonia, as I'm sure you know. I recently was able to participate in Dr. Bhaskaran's study in Minnesota. I did the one-day clinical trial for the wearable collar. I thought it was outstanding. I was so grateful to have the opportunity, and I'm anxiously awaiting that trial to go to stage 2, where you can receive the collar for use, because it's kind of like, oh, here, you could try it, and then you have to give it back. Like if there was a back door, I would have just left with it, but no, not really. I just to mention, I was a high school principal for 27 years, and I'm currently active in local politics. So I'm open about my vocal disorder. A lot of people know. I've shared it. I had to share it on the campaign trail, and that's why I'm so hopeful that there'll be some type of research that will be successful for people like me. And I'm happy to answer any questions.
Sure. Thanks, Risa. Okay, we're going to go right to the questions. Kim, what was a couple of from the previous panel?
Sure. So one of the questions, actually, this is for Dr. Pitman. Could you go into a little bit more detail about the larynx, how it works, and does it work for both AB and AD? And I think that question about whether the treatment works for both AB and AD is one that a lot of our participants would be interested in. And I think most of the treatments are, but I think that's, especially another question we had was what's being done for AB research. So we'll start with you, Dr. Pitman.
Sure. So first of all, the last place is Innsbruck, which is the center. So if anyone out there is in Innsbruck, you're nearby. We think this works by decreasing the activity of the sensory nerve coming back from the muscle into the brain. And when you decrease that activity, you actually decrease the hyperexcitability of the cortex. That's actually exactly how Botox works now. It decreases the excitability of that same nerve, but just indirectly by paralyzing the muscle spindle. And so with electrical stimulation, you can do lots of things to nerves with electrical stimulation. You can excite them, you can inhibit them, believe it or not, you can make them grow. And when we first started looking into, I do a lot of nerve regeneration research, and that's how I came upon the electrical stimulation, because I was talking with Med-El to use their stimulators to actually grow nerves better. And we actually pivoted to this when I came up with this idea. Ultimately, it will work by, it's the same exact thing as a cochlear implant, right? So for deaf children, for the last 20 years, they've been putting these implants in under the skin and running a lead down into the ear. We'll just run that lead to the nerves, feeding the appropriate nerves, feeding the larynx, and then stimulate those. Currently, because most of the patients have adductor spasmodic dysphonia, that's what we're studying. And one of the main reasons for that is we're trying to study very obvious patients. You know, sometimes if you have studies of patients that they don't really have clear adductor spasmodic dysphonia, they may have tremor, they may even have something that's completely different, some sort of dysarthria, and being mischaracterized sometimes of having spasmodic dysphonia, or might have muscle tension dysphonia, then you're dirtying up your data and you might not really have the outcome that you believe that you do. So we're using pretty straightforward, relatively moderate to severe patients with adductor laryngeal dystonia, but there's no reason why this shouldn't work also for abductors in the end. You know, you just have to find which, it's sort of like the Botox, when we give it, you know, which side is better, which muscle is better. You just have to figure out for that patient, which nerve treatment is going to work the best. And it's possible that the internal superior laryngeal nerve will work for both adductor and abductor, and we won't even have to go for another nerve. So I think it's going to work for both. We're also studying cervical dystonia, and it seems to be, you know, helpful for cervical dystonia as well. So it's not specific to the larynx.
Chris, you want to comment in terms of AD versus AB based on what you have learned so far with your DBS studies?
Yeah, sure. So we think that this circuit coming out of the brain is too synchronous, and it's causing spasms of the muscles in your throat. And depending on which muscles spasm, you get the different types of spasmodic dysphonia. There's a lot more muscles that close your vocal cords than open. Adductor is, 80% of all of SD. But the problem originates in the brain, heads down to the throat. And so if you could stop the problem in the brain, and then it would allow the muscles in the throat to act normally. And so deep brain stimulation in our patients, it doesn't matter what the end muscles are doing, it just allows the muscles to act normally. So whether you have AD or AB or mixed, it is irrelevant. It stops the problem at its source.
I think we need to be clear that I think we're all modulating the neural circuit. We're all doing the same thing. We're just accessing it in different places. So I'm accessing it here in the neck. Chris is accessing it in the brain. Russell is accessing in the brain through the skull. We're all actually modulating the same exact network. So there's not that much difference in the end as to what we're trying to do.
Yes, yeah. But using the analogy from focal dystonias of the body, I would preface my remarks by saying, I sure hope we can get it peripherally, because that would be far better than going centrally in the brain, far safer, more palatable to the patients. But in general dystonia, there has been several decades of work of trying to access and block dystonia from a peripheral mechanism. Perhaps you know, but for example, cervical dystonia, patients can often provide a sensory trick to temporarily break the dystonia. It's called a geste antagoniste. It was discovered 100 years ago. And so you can certainly temporarily block focal dystonias using a central, but you can't do it permanently. I hope you can with the larynx, because that would be fabulous. So it's nice to have as Charlie Reavis said, multiple approaches to the same problem, because maybe knowledge from us can help you and you can help us, and then we can find the least invasive method. But for those patients who it doesn't work, I mean, if I could wear a collar and get my voice better, I would buy that collar. But if I can't get my voice better, or if it doesn't last, you know, I would like it stimulated. And if that doesn't work, I think I would like to fix the source. So I think it's a good idea that we have multiple approaches. And I would imagine in a few years, we'll be drilling down on the ones that work, or finding those specific patients. Hey, this is your treatment. You would do really well with the collar. Oh, you should have your nerve stimulated directly by Dr. Pitman. Oh, you know, you really need, you know, DBS or not.
Good comments, Chris. Kim, next question.
I would also like to add, I think one of the most exciting things is that we're sitting here on this panel talking about new treatments. 5 years ago, this was not happening. And so, you know, I think that's the one thing. And we're looking at the brain in all of these different modulations and such, and many of them. And some of the questions that we had received was, you know, since SD is neurological, why are we only treating symptoms? And I can see that there's excitement here that we're looking at stimulation, whether it's internally or externally, of the brain. So we're going to the source. So, you know, I want to thank all of you. We all want to thank all of you for your dedication to finding these new treatments, because it's really exciting. There was a question about study recruitment, and I think it's important to address with all of you. The question was, does being left-handed eliminate me from trial studies? And maybe we can talk a little bit about why treatment studies have these limitations on who they accept. And I know, Dr. Pitman, you've already kind of touched on that, and why most researchers start with AD, but maybe we can talk a little bit more about that. I know I'm left-handed and I got eliminated from being a control. So it's a personal question too, but maybe we can talk a little bit about that too.
Michael, you want to comment?
I'm probably the least qualified of Chris and Russell to talk about that, because it's definitely about central neurologic mapping, and left-handed people have different mapping than right-handed patients, would be is my understanding. And so that throws in a little bit of a curveball when you're trying to really compare everybody with each other. But I would leave it to them, because they're the experts of the central nervous system up there.
Good, thanks, Michael. Russell, you want to talk?
I do.
About
Yeah, I do. We're down regulating, and so our protocols are very, very safe. I also want to mention, though, that we do take very close care to make sure that we're screening participants well enough that we're not taking just anybody, and we're not taking people who might have contraindications for our protocols as well. But no, left-handed, right-handed, we'll take anybody.
Charlie, I don't know if I can jump in, because
Yes.
I spent my whole life studying the brain, and this is really interesting stuff. The left brain and the right brain are very different. Most people, you know, lay people understand that. But for example, music and tonality comes through the non-dominant, typically right brain. So a right-handed person would be left hemisphere dominant. Music's on the right, and most people with SD can sing. They don't have spasmodic dysphonia when they're singing or yelling or intensely emotional, and it's coming through a different circuit in the brain. And so in studying deep brain stimulation, it's also one of our goals is to understand speech. How are we generating speech? We understand language. Language is slightly different from the ability to speak, but our studies have shown that speech is also very lateralized, and that the speech circuit is in the left hemisphere and a right-handed dominant. What we are doing is all our patients are having, they call it functional MRI, and they talk, and we can see what part of the brain lights up and is handling their speech, so that we can target the specific side that has their speech dominance.
Yeah, just if I could follow up with you guys on, and I've noticed a couple of questions, and then the last panel there were a couple of similar questions. If people are on certain types of medications, like antidepressants, for example, do you eliminate them from your study, Chris?
No, a lot of patients are on antidepressants. And I would say, I would preface my remarks by saying we are looking for severe SD. We are not looking for mild SD that, well, maybe you got a little bit of it, or a little vocal therapy will help you, or Botox is working well. We're looking for the patients where that has failed. And so our patients are typically the worst of the worst. And so, yeah, I mean, they've all got depression. They're all on medications, and that's who we want to treat.
Okay. Michael, how about with your project? You're muted.
Keep doing that. They just have to have a significant amount of spasmodic dysphonia symptoms. That's it, and be over 18 and under 65. That's
Russell, how about you guys?
Yeah, that's the same for us. We're recruiting between 25 and I believe 70, 75, 21 and 75 we're looking at. As far as medications, only if they're on certain anti-seizure medications, right? Those are the people that we're excluding, and generally we'll even consider those people if they haven't had a seizure within the last 2 years.
Okay. Diva, how about with you guys with your neuromodulation or vibration? Do you guys eliminate potential candidates?
Yes. So being on antidepressants or anti-seizure medications is one of our criteria for eliminating, because we are looking at EEG, which is a map of the whole brain activity. So those medications do have an effect on EEG. And so that is just one technique to, again, like we're comparing that value with everybody in the study. So we want to make sure we have enough of the participants with normal EEG data so that we can compare all spasmodic dysphonia together. Again, this is more of a criteria based on recruitment to get enough subjects that we can compare together. If we had a large number of participants who all had EEG, antidepressants or other medications or a stroke, we would just categorize them into one group so we can compare within that group. But because we're not getting enough participants, we're trying to not have that group.
Okay. That leads me also to something I know that comes up frequently. We get a lot of questions, and I'll ask each of you to comment on. And Diva, while you're talking, during the pandemic, obviously you could not recruit. So could you give us a status on where you guys are at the moment and what your plans are for over the next few months for recruitment?
So yeah, one of the main study that is going on is a long-term 11-week study. So which was cut short because of the pandemic. So we didn't have a lot of people complete the study who were in the middle of the study, and then we couldn't recruit because people who wanted to travel and participate could not travel in the pandemic. So our initial goal was to get 40 participants by this June, I guess, but we are not. I think we're 15 to 20 ongoing, and again, because it's a long-term 11-week, tracking people for 11 weeks is not easy with people dropping off or not traveling and things like that. So we are 15 to 20 participants with the AD doctor clinical trial, and the collar study, which looks at AB doctor laryngeal dystonia, was supposed to start September 2020. Again, got delayed. We just started January 2021 or December, January. And so we have had, luckily we have had people traveling and able to visit. So we've had, I think, I want to say 8 participants so far. And we have 2 more or so in line to participate in the upcoming months.
Okay, good. Michael, how about you? What's been the impact of the pandemic on your project?
Yeah, so we were doing pretty well. Then it stopped. Then we recruited 2 more patients, and then it stopped again. And so, hopefully things will open up and loosen up and start. It's a pandemic that waxes and wanes in different parts of the world. So it's a little bit difficult And I did want to say, actually, there's one other exclusion criteria that's important. Because we're stimulating, you can't have an active other stimulator in your body. So that would rule you out. And obviously some other CNS disorder, you know.
Yeah, that, Michael, that's a good point, because I've had a few people mention the question too, if they had a pacemaker, would that eliminate them?
Yeah, no other stimulators.
Yeah. Chris, how about you, the impact of the pandemic?
Yeah, like everyone else, we were hit, but the timing is such that we just finished our first randomized controlled double-blinded prospective crossover trial, and it's just been accepted for publication. And now we're in that moment where we are starting to recruit for our randomized controlled trial phase 2 prospective randomized double-blinded crossover trial. And so we're in the process of recruitment. But we are fortunate to be in Western Canada, where COVID is not as bad, but we're the only center. So we have a lot of patients who are potentially interested.
Okay. Russell, how about you guys?
Yeah, I'll just say, Dr. Honey, I just came from Toronto just about a year ago, and boy, it was, I'm sure, very different there than it is in Vancouver. Yeah, at MGH, we are recruiting. We have a lot of protocols in place, both lab-specific protocols, but also MGH-specific protocols for dealing with we also have an MRI component to our study. So how many people are allowed to be in the MRI suites at a time? How many people are in the treatment rooms at a time? So we're taking it very seriously, but we are recruiting. We're actively recruiting, and hopefully going to be doing so for the foreseeable future.
Good, good. I don't know if Kim, are you back? Yeah, we've had a little problem with the internet, and Kim's having some issues. So let me jump to Risa. You want to talk a little bit about your personal experience with the vibration project and wearing the collar, and what the effects were, and how long those effects lasted for you?
Sure. The actual study itself, the researchers were excellent. The protocol was excellent. I traveled in March, which was, I was lucky vaccinated, but it was still a tough time for travel. And I mentioned that because if you're thinking about participating, I can tell you that in Minnesota, everything was done very well. It was just a few hours. I had the collar, which I thought was comfortable. And I don't know who mentioned it, maybe you did, Chris. I would wear that collar and I wouldn't care. Like, if you could give me that, I wouldn't mind. It was unobtrusive. I also had the EEG, which was weird, in room with the cap and the collar. And of course, all the famous sentences that all of us with ST remember that you have to repeat when you're either getting Botox or getting an assessment, and you would look at these sentences on the screen and be like, oh my God, not that sentence, I remember it. And then, you relax and you do your best. I left. It was definitely probably one of the first times I felt, wow, like I feel hopeful. I've tried lots and lots of different things Botox. I had an excellent doctor. It just didn't really work. It didn't suit my lifestyle. I've done a lot of different things, but I left Minnesota feeling like, wow, there's hope.
Excellent, excellent comments, Risa, and absolutely, and you know, speaking for our entire organization, the work that all of the researchers are doing on this panel and other researchers as well is just incredibly exciting. I know I go back a few years ago with some of the conferences with Michael, and boy, you know, what's happening now compared to, gosh, 7, 8, 10 years ago is just incredible. So, you know, as our organization and the whole community, we can't thank you guys enough for these projects. And I've noticed a couple of comments about people wanting to know more about each of these studies and projects. So I would encourage you to go to our website, dysphonia.org, and we have up-to-date information on these various projects and ways to participate, and the ones that are starting to recruit again. And as a couple of the other projects start to recruit here in the US, like the Michael's project, we will get all that information out to you guys just as soon as he's ready to flip the switch. So check our website, dysphonia.org, for updates and all the information. Michael, could you just talk a little bit about, you know, the procedure itself? I know I've had several people ask me about, well, geez, you know, what does that really mean? How's it really implanted, and those types of things? So this may be a little bit about the procedure itself.
Yeah, so for that, we would reflect more on what we do for cochlear implants at this point. And also, MED-EL has implanted 9 of these specifically for bilateral vocal fold paralysis. And the implant is a very small battery. If you've ever seen a person with a cochlear implant, you can't see the implant, but then they put a battery on top of their skin. The actual stimulator is underneath the skin behind the ear. And they've worked that out, putting it here for like 20 years and figured out how to do it through a small incision with low risk of infection. In the beginning, cochlear implants were a huge incision, lots of infection problems, but you know, thankfully other people have been doing something similar. So if we decide to put it here, that's really all worked out. And you didn't just take a lead that would come down under the skin and go to wherever we feel the area of stimulation would be the best. Okay, so it would all be implanted, you know, no major things. Then what you would do is, we think the stimulation would be about 30 minutes. So you would just take a battery and just put it on here, or if it's, some of the implants for the larynx are actually right here on the sternum. You eat your Cocoa Puffs for your breakfast, because it doesn't hurt and there's no pain or anything like that. And then you take it off and you put it away. And what we found, and also even Tanazi in hand focal dystonia, if you stimulate for a few days in a row, you might get a couple weeks or more of good function, good voice function. And even the crude stimulation in our patients of just putting a hook wire electrode into the vocal fold, patients improve for 3 to 12 days. So the hope is you'll be able to get repetitive stimulation, will get longer and longer durations of benefit. And even if you have to put it on for a week, twice a month, it's really not much of a big deal, because the goal is, instead of having this up and down of Botox and having to come into the doctor's office, you pretty much never have to come to the doctor anymore, and you treat yourself and you just sort of keep the voice nice and steady. And as you're finding a decline, you just start to stimulate yourself again. So that's the vision of what this is going to be.
Okay, excellent. Thanks, Michael. Russell, you want to talk a little bit about the process and the procedure that you guys are using?
Absolutely, yeah. So this most recent study is a 5-day trial. So we're actually bringing, it's 2 phases. The first phase You come in, you receive the RTMS, the repetitive transcranial magnetic stimulation treatment. On the first day, we do some neuroimaging and MRI of your brain, and then a functional MRI as well. And then you have 5 days of this 20-minute procedure. You're sitting, there's just a small coil that lays on your head. And you're stimulated for about 20 minutes, again, at a very, very low frequency. And it essentially just feels like someone's just tapping on your skull. It's really, it is non-invasive in that way. You come 5 days in a row. On the 5th day, we reapply the measures that we did on the first day, which are just voice measures and kind of some acceptability and feasibility measures as well, and tolerability measures to see how that 5-day stint went. You have a 3-month period where you don't receive any RTMS, and then we bring you back for another 5 days, and you either receive again the RTMS or a sham, depending on which one you received in the first study. So you may have received a sham for the first week of our therapy. In that case, you would receive the actual RTMS therapy, and it's exactly the same thing, sounds exactly the same. We're just not stimulating the area of interest. So it's a 2-phase study, 2 weeks. We do offer compensation for those who have to travel to us, because we know that is kind of a difficult thing as well. But we do, because we understand that it is kind of difficult to come to Boston for 5 days in a row. We do offer compensation for travel as well as compensation for the study. So.
Good, thank you. I have an interesting question that someone asked me, as a matter of fact, a couple of days ago, Russell. And with the project and the stimulator that you're using, do you actually shave a person's head for certain areas of where you put this device on their head? I hope I gave the right answer, so I'll let you answer it correctly.
For me, that wouldn't matter because I've been nearly bald for about 10 years. For others, that would matter. No, we do not shave the head, which is great, right? There have been some studies that say that depending on the thickness of the hair, you may adjust the distance between the coil and the cortical surface of your brain, which can have an effect. But no, we do not shave people's head in order to get closer to their brain. It's again, I would consider that more invasive than non-invasive if I had to shave my head. So no, we do not shave people's heads. Keep your hair.
Good, thanks,
Yeah.
Thanks, Russell. I appreciate you validating my response to that person.
No, thank you for telling me that. I appreciate it.
Chris, you want to talk a little bit about, I mean, we all, or a lot of people, hear DBS, been around for a long time. It's been used for Parkinson's, generalized dystonia, and a couple other conditions. Maybe just, again, talk a little bit about the process of actually doing DBS.
Yeah, so that is my world. And actually what happened is about 50% of patients with spasmodic dysphonia also have tremor. And there were some patients who had very bad tremor, happened to have SD, and they came for deep brain stimulation. That's just another Tuesday for me to do that surgery for tremor. And then serendipitously, they said, oh, by the way, my voice is normal again. And so that was really the source of the concept that we could go centrally and block SD. And then we used that data to support just going after people who had SD, but not with tremor. So deep brain stimulation is a brain operation where an electrode is placed in the brain to modulate the part of the brain that's not working. And we use it to block tremor, to block other forms of dystonia, more bigger generalized forms of dystonia. Perhaps it's most commonly used in Parkinson's. I've done about 1,000 operations. We do almost 100 a year in Vancouver. That's a little unusual. That's a very large center. And about 250,000 people around the world have had deep brain stimulation.
Okay, great. Divya, you want to talk a little bit about, again, little more in depth about the process that you guys are using with the collar and exactly what that means and what you're attempting to do with that stimulation.
So it's, again, goes back to the same thing Dr. Pitman mentioned, is we're all trying to attack or go through the same pathway where the vibration that we apply, we plan to activate the mechanoreceptors in the muscle spindles, which goes to the somatosensory cortex and motor cortex communication. And again, that communicates with the laryngeal muscles. So it's all trying to modulate the neural mechanism that's going on, but again, through a surface and through the laryngeal muscles is what we are trying to activate as well. So we apply 2 vibrators. There are the sizes of AAA battery size, and we just place them on the muscles right there, and we vibrate it. And so it's all on top of the skin surface. It's not invasive. And so that's about the process that goes on. We vibrate it, and then we, like Risa said, we have them speak the different, speech assessments, sentences that activate those vocal cords. And then we also look at the brain activity and the EEG. The other thing I don't know if I clarified before is we do disqualify people who take antidepressants and anti-seizure medications. We don't think that the vibration might not work for you, but we cannot look at the brain activity of those people to understand how that vibration works. So that is one of the factors that we look at, but does not mean that the vibration might not work for you.
Okay, excellent. Okay, I noticed in our chat, lots of people are commenting and looking for more information and contact information from all of you and your projects. And again, I would just encourage all of the participants to go to our website. That's the easiest way to get the information, and we try to keep that updated. And thanks to all the researchers for providing us up-to-date information. And so again, go to dysphonia.org and you'll find a lot of that information. And if you have specific questions, we can certainly contact the researchers directly and get that information for you. So
Charlie, I'm back.
you're back, okay.
I had some issues, so some computer issues.
Right.
We've got a few more minutes, so there are a few more questions, and maybe
Okay.
I'll address some of them. One of the question is, are the DBS trials only available for Canadians, or can Americans join too?
You can, that's plus or minus. You'll have to go to stopsd.com and just communicate with us. Just shoot us an email and we'll go over the details. I did want to say something unrelated and more generalistic, and that is this meeting has evolved dramatically from basically accepting Botox as the treatment forever, and nothing against Botox, it works for a lot of people, to demanding other treatments and understanding the cause of SD. And because of Charlie and his drive and the members' donations, all of a sudden there's a pool of money for researchers like us who are interested in understanding those questions. And I just want to say it's your members that have driven this research, and this meeting is very exciting because maybe DBS will work, maybe it'll pan out, but the answers are coming, and it's very impressive listening to Dr. Pitman, Dr. Banks, Dr. Bhaskaran. That's all exciting research, and it's because of Charlie and your members, and I congratulate you. There was a pool of money that enabled us to do this research because you guys, gave it to us.
Thanks, Chris. I appreciate that very much. And again, it's, it's just fantastic for us as a community of people with voice disorders to have dedicated professionals like all of you who are interested and absolutely committed to pursuing some of this research that we're talking about today, which is, as we touched on a little bit earlier, you know, just a few years ago, I mean, five years ago or so, we would not have been talking about really any of the things that we're discussing here today. So, Kim, yeah, another question?
Yep, there's another question regarding there's some success with the PONS device currently being manufactured in Canada, but developed in Minnesota. Is any study going to be done to qualify it here in the US for SD? Not sure if anybody can address that.
I don't know, Chris, are you aware of that?
Yeah, is that the one you put on your tongue?
It doesn't say, so I'm not sure. It said it was a PONS.
There's a peripheral stimulation that's put on the tongue. It's being used for a variety of things. Some of the problems with that is there's a massive placebo effect. And you know, people desperately want to get better. They know that this device is on. They've paid money for this device, and you know they want to get better. So I'm just waiting for the studies to come out where you truly, and I mean, it's hard to randomize someone who can feel that they're stimulating. But all the studies, all the studies that you heard today have to be validated. Is it work long-term? Is it more than just placebo? Were you really dealing with SD and not conversion disorder? And all of the researchers here today are working towards proof of long-term efficacy with their device.
Great.
David, you have any information about that or are familiar with it?
No, I have not heard of the PONS device. As far as I know, I don't know who would be working on that device from University of Minnesota.
Yeah, Michael or Russell, either one of you have any information on that?
No, but I do echo what Chris said in terms of it's really important for us to, you know, prove that these things work and weed out those placebo effects, etc. You know, and not only in one center, but in multiple centers, right?
I just Googled it.
I did too. It does everything.
Yeah, so it's a portable neuromodulation stimulator, and it vibrates the tongue, and it's been used to try and help walking in multiple sclerosis. And so before you go any further, multiple sclerosis is a disease that comes and goes spontaneously, and any therapy that happened to be given right before you were going into a remission, the patients will swear that it worked. And that's what happened with the Liberation device. And so you have to be very careful with people who are marketing devices that it has been peer reviewed. And like Michael says, multiple centers have proven it scientifically. And I think the National Spasmodic Dysphonia Association is lucky to have researchers who are dedicated to doing exactly that.
That's good information. And hey, talking about real-time investigative analysis here, boy, we're right on top of it. Thank you, guys.
Okay, this one is actually for Russ. Is RTMS similar to what they're doing for depression? That is a 6-week daily thing.
Exactly the same, just a different area of the brain. So they're targeting the motor areas of the brain as opposed to the areas of the brain responsible for controlling and modulating depression. Yeah, same exact thing, and similar dosage as well, at least for our initial study. Yeah.
Perfect. Question about essential tremor, and does it disqualify you from these studies?
For us, for now, it does.
We'd have to know more about the tremor, but I would assume it may be difficult to have you participate in at least in this initial study. Yes.
Okay.
And
I think it's important. I think it's important. Russell's saying, like, just because we're, you know, the initial studies, we want them so clean, you know, that's why they're getting excluded. But don't get discouraged because those patients will eventually come in under, you know, future studies. That's important to understand. Yeah.
Good
well,
point.
and also, sorry, Chris, I know you're just about to talk. Sorry, I apologize. Our protocols require that you remain relatively still too. So this is a fixed coil on your head. We'll come out with ways to attach it to your body so that it doesn't matter if you move, but right now it's a fixed coil on a chair. And so if you're moving, it'll affect where we're stimulating, and our results will be contaminated a little bit in that way. But we are working on that for sure.
And I would have to double check also if it's a central tremor of the hands, you might be able to be in the study, but certainly not of the larynx. If you have tremor, you're not you're excluded from this study.
Okay.
Just
Diva, for you guys.
Just a note to Russell, essential tremor patients only have tremor when they're activating. If they're sitting or lying, they don't shake. So they can sit on their hands and they won't shake your head. But the problem for deep brain stimulation is that the deep brain stimulation stops the tremor. So you can't blind them. They know instantly when the stimulator is on. You can't feel the stimulator, but if your hand stops shaking, you know it's on. So they are unblinded. And so we have excluded them for that.
The same.
Thanks, Dr. Honey.
Yeah, the same. We also don't include people with tremor. Again, this goes back to research being not, it's more a scientific approach, and versus real life. Real life patients have different other conditions as well.
Well, it's just what Dr. Pitman says. You want the purest possible cohort to do your first study, and then proof of concept, then proof it's reliable, and then you can spread out, because we know a lot of SD have tremor, right? It's a very similar circuit.
Right. Okay, I think I again appreciate all of you on the panel. Great discussion today, and fantastic and exciting work all of you are doing. Thank all of the participants for all of their questions and their interest, and so it's been fantastic. We will do a quick roundtable here, and for each of you to give wrap-up comments. So, Diva.
I think I echo everybody, Dr. Honey's comment as well, where NSDA has been a big proponent for the research, and also helping us, guiding us, telling us where the problems of the patients are, what is a good avenue to look at, and I think it's been a good working collaboration to work with NSDA and your members to understand what type of research is needed versus, this is what I have and this is what I want to do, versus the patient's feedback and getting feedback from people who actually have the disease and who suffer from it long term, to know and understand what is needed for them. I think it's been a great perspective to have while conducting research. So,
Excellent. Thank you, David. Chris?
I'd just like to say thank you, Charlie, to you and your organization for supporting this work. I think it's important not to be complacent. I'm stuck with SD. My treatments don't work very well. No, I think you need to demand more as stakeholders, and you have a team that is working to try and figure out how to help people today. But what's particularly interesting for me is, in trying to help the people today, we are beginning to understand the neurophysiology of speech. And that is super fascinating for us, and may have ramifications in other areas. So thank you for demanding attention. There are a lot of rare diseases out there that are being ignored, and it's people like Charlie that's pushing the profile of SD to attract people like us, who then get hooked on meeting patients like you.
Great. Thanks, Chris. Appreciate it. Russell?
Sure. Yeah, I'll fourth or fifth the sentiments from everybody. Thanks so much. This has been a really, really awesome experience. I'll just say too that I think, as has been mentioned, I think one of the reasons that we've come so far just over the last 5 to 10 years is not just because of the improvement in technology and things, but also because I'm a speech pathologist working in a physical therapy lab that's in an Institute of Health Professions at one of the largest hospital networks in the entire United States, that has neurologists, neuroscientists, occupational therapists, the whole run of people working together on these problems. So as technology is getting better, we're also combining forces, and I think that's wonderful. And I think it's mechanisms like the NSDA that allow those connections to happen. And we're super, super grateful. And that's on behalf of the BRL and also Dr. Kimberly.
Great. Thanks, Russell. Michael.
So again, Kim and Charlie, thank you for just supporting us and supporting, you know, the NSDA. Really, you guys are what are the engine. And this is always a very informative day. Right now we've got 4 people who've got 4 different ideas of how to treat this. Hopefully in 5 years we'll have 8 people, and the panel will be too big, you know. I do look forward to that. And I did notice that a lot of the questions were about, you know, how can I get in the study? What are the inclusion criteria? What are the exclusion criteria? You know, and it's hard for us to remember off the exact top of our heads, to be honest, of every inclusion and criteria and exclusion and criteria when you write these studies. So those were the general, I think everyone gave you, you know, the sort of the big hits of who can come in, who can't. But I would say don't be discouraged by that. You know, if one of these or all of these studies interest you, you should reach out to everyone. I think through the feed, everyone's put their contact information for me. I'm at voice@columbia.edu. That's our Center for Voice and Swallowing. And even if you're in the US and you can't be in the study now because it's happening in Europe, we can keep your name and then get back to you as we need to. So that way we know, you know, who's at least interested. And maybe when the time comes, you will be, you know, allowed to be in the study and welcome to come. So don't focus on, you know, whether or not the study's right for you today or tomorrow. Just if it's interested, get in touch with the investigators and we'll get you on that list, and make sure that Charlie and Kim also know. So, because each time we move, we reach back out to them. We say, do you have people who want to try this? So they're almost like a clearinghouse for this, right? And they're a great resource and they know what's happening. So I really think, you know, just reach out. Don't be shy.
Good. Thanks, Michael. Appreciate it.
If I could just,
I would just like to thank you, Charlie and Kim, and our medical professionals, and also I'd like to thank my peers in our support groups on the NSDA website, because this, it can be very isolating if we let it. But thanks to all of you, we have options.
Great. Thanks, Risa. I appreciate it. And I want to just quickly add a comment to what basically all of you said. And, you know, one of the roles that we play as the NSDA and as an organization is to really get researchers together talking, whether it's in a symposium or on panels like this, but in also ways that some of the research teams can collaborate with each other, because there's so much of this work that various teams are doing that can help another project with somebody else working on a specific approach to it. So that's another role that we play, and we've been doing a lot of Zoom calls here in the last few months with various research teams and making them aware of, hey, by the way, you know about this researcher or this team working on whatever particular project. So again, I appreciate all of you, all the work that you're doing, and it's so exciting and fantastic. So keep it going, guys, and thank you very much. Kim, you want to comment?
Yep, before we conclude with this panel, 2 comments. We actually on the website keep a list of, under our research section, of studies that are currently going on. We obviously have some updates to make. So I know, Russ, you are just starting to recruit with the AB and such, but I put the link in the chat. And I'd also mention is that oftentimes when researchers are recruiting, they'll ask us to send out email blasts, and sometimes it's geographical, sometimes it's wider. So you may see email blasts coming through saying that they're being recruited, and we try to keep all of that information current. You know, I just want to thank all of our researcher and Risa for providing the other perspective, because without you and people in our community, these research studies don't happen. And especially with the AB ones, we've been very integral in pushing our researchers to look at AB, and we appreciate their willingness, because it's been a recruitment issue or a recruitment concern, and we say, not a problem, we can get you the patients. So, so I just want to thank you all for that.
Thanks, guys. Appreciate it so much. Take care.